Let's briefly talk about what happens to the amino group in a situation where there's high amino acids. We talked briefly before about the urea cycle and how that's a detoxification pathway to sort of sequester amino groups in the form of urea. Now, in a situation where you have excess amino acids from high amounts of proteins that get chopped up by proteases in the gut, that amino acid can have a source of amino groups which needs to be immediately remedied by the cell. So the liver specifically has a lot of these enzymes known as transaminases, and transaminases actually convert an amino acid to its corresponding alpha keto acid. They are in an equilibrium. Usually the equilibrium has a constant of 1, so the transaminase does not favor the reactants or the products unless there's some sort of shift in the metabolic flux, and an influx of an amino acid via dietary consumption can be enough to shift the equilibrium towards the formation of that alpha amino acids corresponding alpha keto acid. So in the cell, we basically have the amino acid that's sort of the designated nitrogen or amino group storage vessel, and that is the amino acid glutamate. So here we have transamination 1, again catalyzed by transaminases. They take an amino acid and it donates its amino group to the alpha keto acid, alpha ketoglutarate. So the chemistry involves the formation of a shift base and a cofactor known as pyridoxal 5-phosphate. We'll briefly touch on the mechanism a couple of slides from now. But here you can see the alpha amino group sort of amidating the C double bond O through a shift base mechanism. As a result of that, this amino acid becomes the corresponding alpha keto acid. We've talked briefly about some alpha keto acids and their corresponding alpha amino acids. For example, alpha amino acid for aspartate, its corresponding alpha keto acid is oxaloacetate. So like that, all 20 or so amino acids have their corresponding alpha keto acids generated via these transaminases. So when the amino acid donates its amino group to alpha ketoglutarate, it itself becomes this amino acid known as glutamate. Remember, we talked about glutamate as sort of being the central hub or the central reservoir of alpha amino groups. And then the alpha keto acid is formed from that corresponding alpha amino acid. So recognize what's in that red box. The alpha amino group has now been shifted to glutamate. Now that glutamate can actually has one of several fates. One of the fates is it could react in another transamination reaction. I call that transaminase number 2. The glutamate can be subject to oxidative deamination. There's also in the cell something known as glutaminase, an enzyme that converts glutamate to glutamine. And there's also glutamate synthetase. So in the next slide, you see the transamination of glutamate from transamination 1. This time it donates its amino group to oxaloacetate. In turn, that glutamate becomes alpha ketoglutarate, so we regenerated the alpha ketoglutarate so that transamination number 1 can happen. So this can be essentially a circuit, a metabolic circuit. And that oxaloacetate, by accepting that amino group, gets converted to aspartate. Now that aspartate, as we've seen before, can donate its amino group to urea via the urea cycle. Take any amino acid and it's going to react with the transaminase and it will give you the alpha keto acid and the corresponding amidated form of the enzyme. Now pivotal to these transaminases is the cofactor pyridoxal 5-phosphate. This pyridoxal 5-phosphate is a precursor for vitamin B6. It's a vitamin B6 precursor. And that's really going to provide the shift base for which you get the amidated enzyme. Now this amidated enzyme, through the collapse of the shift base, will donate its amino group to an alpha keto acid. Usually it's alpha ketoglutarate, and you regenerate glutamate. There it is, that amino acid. That's the central nitrogen or amino group reservoir or primary storage component. So the net effect is you have any amino acid getting converted to as alpha keto acid, and that amino group gets transferred and stored as glutamate. Very, very critical that we understand that glutamate just has picked up that amino group from the alpha amino acid. So noteworthy in this is that the alpha keto acid doesn't necessarily have to be alpha ketoglutarate. It can also be alternatively oxaloacetate. In turn, the alpha amino acid can be aspartate. Let's talk briefly about pyridoxal 5-phosphate. This is an important cofactor in transaminases. This is the form shown here in part A that actually converts the alpha amino acid to its corresponding alpha keto acid. And the boxed form in part D is the amidated form of the enzyme, and that form actually converts the alpha keto acid to its corresponding alpha amino acid. So these transaminases, if I can go back to the previous slide, these transaminases go in both ways, and dependent if you have an amino acid enriched environment, you're going to make the alpha keto acid. If you have an keto acid enriched environment, you're going to shift the equilibrium more towards generating the amino acid. So returning back to this slide, this is the form of the cofactor that actually favors the formation of the alpha amino acid, and this is the form of the cofactor that favors the formation of the alpha keto acid. All of these function via a shift base. So the shift base is very important. A lysine residue is actually also very important. It conjugates the pyridoxal 5-phosphate to the active site of the transaminase. So lysine is going to be a very important residue that sort of is attached to this cofactor, this vitamin B6 cofactor. So going back to two-step transamination, one of the products is glutamate. It could donate its amino group to oxaloacetate, or it could be subject to another important process of amino acid metabolism, and that's oxidative deamination. So this is a straight-up redox reaction. The carrier of the reducing equivalents can be in the form of NAD+ or NADP+, and you get the reduced form of the cofactor. What's important here is you've essentially stripped the alpha amino group of glutamate. Remember, that came from the amino acid that was transaminated in the previous slide, and it's the primary reservoir and storage component of alpha amino groups. What has essentially happened is that this amino acid glutamate has had its amino group stripped, and you get the ammonium ion. And NADPH is produced also. This enzyme is highly regulated. It is found in the mitochondrial matrix, so that is no irony there because the urea cycle enzymes are also found in the mitochondrial matrix, at least 2 of the 5. What's very interesting is the whole repertoire of allosteric effectors. You can have negative allosteric effectors, such as GTP and NADH, that tell you that the cell is essentially energetically fulfilled and all the reducing equivalents are reduced. And also, oxidative deamination is linked to fatty acyl coenzyme, fatty acyl coenzyme A levels. It is also linked to steroid levels. These molecules actually allosterically inhibit oxidative deamination, and they inhibit glutamate dehydrogenase. Low energy states such as ADP and NAD+ allosterically activate it. The amino acid leucine allosterically activates glutamate dehydrogenase. So oxidative deamination and glutamate dehydrogenase is something that's very important. This stripping of the alpha amino group into the ammonium ion is now ready to go into the urea cycle. So here you see asparagine getting converted to aspartate, and asparaginase actually strips this. Now, this is not an oxidative reduction reaction as you saw before in the previous slide with glutamate dehydrogenase. This is just more of a hydration reaction. You can see water sort of splitting this bond. We saw a similar mechanism with arginase. So arginase does kind of the same thing. Here, asparaginase is going to hydrate this bond right here, cleave off an ammonium ion, and that ammonium ion can go get incorporated into the urea cycle. The asparagine that is deaminated via asparaginase gets converted to aspartate. That can participate in the urea cycle, or that aspartate can participate in gluconeogenesis. Now, as well as an asparaginase, there's also a glutaminase. The glutaminase is going to take glutamine and convert it to glutamate. pretty much the exact same mechanism as seen here. Glutamine gets converted to glutamate, water comes in and strips that amino group. It's a hydration reaction. I want to focus the attention here on the middle column and then that third column. You can see transamination to aspartate, but asparagine synthetase actually converts that aspartate back to asparagine. This is exactly the opposite of L-asparaginase that you saw in the previous slide. So if I can go back to the previous slide, you have asparagine getting converted to aspartate via asparaginase. Now coming back to this slide, you have the exact opposite. You have aspartate being converted to asparagine, and the enzyme that does that is the asparagine synthetase. So two enzymes, each catalyzing the reverse reactions. Same thing occurs here with glutamate and glutamine. Glutamate can get converted to glutamine. The enzyme that does that is glutamine synthetase. That's an ATP-dependent process. And you get glutamine. Now, a glutaminase is going to do the exact opposite. The glutaminase is going to convert glutamine and convert it to glutamate. That in an ATP-dependent manner, it takes ammonia, or the ammonium ion, and makes glutamine. And it hydrolyzes ATP to form ADP and phosphate. So it's an energetically, so this reaction requires energy in the form of ATP. So many different feedback inhibitors for this highly sensitive enzyme. It's regulated, as stated before, by adenylation, and it's regulated by up to 9 different feedback inhibitors. Now, the adenylation enzyme actually will adenylate a tyrosine residue and lower its activity. The actual adenylation of the enzyme glutamine synthetase itself is regulated. So alpha-ketoglutarate and ATP actually stimulate the enzyme that will adenylate glutamine synthetase and thus lower its activity. Glutamine and inorganic phosphate actually feedback inhibit and lower the activity of the adenylase or the adenyltransferase that will adenylate this enzyme. So the glutamine and inorganic phosphate sort of inhibit the adenylation of the enzyme, or the enzyme that catalyzes the adenylation, and alpha-ketoglutarate and ATP stimulate the enzyme that adenylates glutamine synthetase. So in conclusion, there's a number of different avenues and mechanisms to strip the alpha amino group for preparation into the urea cycle. We've talked about two-step transamination, where the amino acid donates its amino group and eventually gets in the form of glutamate. There's oxidative deamination in the form of glutamate dehydrogenase. And finally, we've talked about the formation of glutamine, as well as glutamine synthetase play a role in regulating the amount of amino groups that can be formed or get incorporated